right, ladies and gentlemen, thanks so much for joining in. This webinar is hosted to you by GRCTS, also known as GRC Training Solutions. My name is Alvin, and a quick introduction about our expert today. His name is Dr. Ludwig Huber. Ludwig Huber is the Director and Chief Editor of LabCompliance.com, the global online resource for validation and compliance issues for laboratories. Mr. Huber is an expert for FDA and equivalent international compliance and for ISO IEC 17025 laboratory accreditation. He's also the chairman, presenter, and panel discussion member at U.S. FDA industry training sessions and conferences. He has given more than 300 presentations, mainly in the areas of GLP, GMP, 21 CFR Part 11, and validation globally. He's also the author of the books, Validation and Qualification in Analytical Laboratories, and Validation of Computerized Analytical and Network Systems. Without further ado, Ludwig, you have the complete control, so you can begin the meeting when you're ready. Thank you.
Oh, okay, thank you very much, Alvin, for introducing me, and thanks everybody for joining us today to this presentation, which is on validation of analytical methods and procedure. And we will talk about a relatively new FDA guidance document that came out already back in 2015, but it's a very important time because at this point in time, FDA is just starting to enforce this guidance document. So it's a very good idea to listen to my presentation. Okay, so I will just now continue, and this is the agenda of today. And I will start with a brief overview and make a comparison between this new guidance document and the one which was previously there, which was already has been introduced in 2000. Then I will go through its background, the objective, and the scope. I will go then through individual chapters, starting with a chapter on analytical methods development. Next, I will talk about documentation requirements for analytical methods. I think this is also very important. And then the biggest chapter is on validation of non-compendial analytical procedures. These are methods developed in-house. Then I will continue with verification of compendial procedures and FDA thinking on using, validating alternative procedures. So this is also very important. Then next I will talk about requirements for transfer with the third last bullet here on method transfer. And the chapter of lifecycle management is new, and it also specifies requirements for revalidation and review of methods. I will close the seminar with a discussion on post-marketing changes and how FDA verifies analytical methods as validated or verified by the industry. And I think Alvin already talked about, if you have any question, you can, we will have a Q&A session at the end of my presentation. You can enter your questions in the chat box already, or we also will open up the phone line later on for allowing you to ask verbally the questions, which I can answer then right away. I think we now talk about the FDA guidance on analytical method validation. And I have here the things which I consider are new. So, for example, it really heavily promotes what they call the life cycle management approach. It has chapters on method development with focus on robustness testing. So that's important because robustness testing, I mean, this is probably becoming more and more important. And this also will be done during development, this method development already, so I will talk about this. It requires submission of method development data. I mean, this is also something new, which we have seen. It refers to ICHQ2 for validation parameters and tests. That is also very interesting, very important. This guidance document from the FDA is not very specific on what to do and how to test it, what parameters to test, and what will be the test procedures. No, FDA refers to ICHQ2, which I think I also consider this as a golden standard for analytical method validation, which is proven that is really referenced in this FDA guidance. Also, I think everybody was expecting, or most people were expecting, that this guidance document from the FDA will say something like, we should now follow the principles of quality by design. No, this is not what it says, but it includes requirements which we know from QBD, quality by design. So it very frequently mentions risk assessment. It has chapters on verification of compendial method. It has chapter on method transfer. And it also includes a chapter on alternative methods. This is, for example, if you don't like a compendial method, then you can develop an alternative method which may give the answer better, which may have better precision. So I think FDA is also very supportive to look at this guidance, to look at this reference material, which is also very important here. So now we talk about, definitively no question at all, this guidance document which I just talked about, which is the topic of today currently, is the most important one. It's shown here in the second bullet, analytical procedures and method validation for drugs and biologics. There are other ones which are very important. I already mentioned ICH guidance for industry Q2, and this is both talks about the text of the guidance and also the methodology. Then we have other FDA industry guidance, like the one on bioanalytical method validation. There is one on validation of compendial methods, verification of compendial methods, and the transfer of analytical methods, which are both USP 1225, 1226, and also 1224. So this is also important. So I think this is now the table of contents of this new guidance document. So we talk a little bit about it. It has an introductory chapter with objective and the scope. The background information refers to FDA regulation. Then there is a chapter on analytical method development, followed by requirements on what analytical procedure should include. Examples are the principle and the scope of the method, the used equipment, and also operating parameters. There is a chapter on requirements for reference standards and materials, which is also very important. In addition, there is a chapter on validation of methods for new drug application, for abbreviated new drug application, biologic license application, and drug master files. This has recommendations also for non-compendial as well as for compendial methods. The chapter on statistical analysis and models is more detailed than in the existing guidance, and new is also the chapter on life cycle management of analytical procedures. Again, I repeat, life cycle management of analytical procedures with recommendations for revalidation, for periodic review, and comparability studies, and it also has recommendations for reporting post-marketing changes to an approved new drug application, ANDA, or biologic license application. So this is also all very important. I think I'm talking about the scope of the guidance now, and first of all, the guidance replaces the existing draft guidance, which has the same title, right? But that one was from the year 2000, and it also replaces the FDA guidance currently called submitting samples and analytical data for method validation. That was also relatively old, from 1987. The new guide applies to drug substances and drug products for abbreviated new drug applications, and for biologic license applications, so it's quite universal. The guide also recommends to consider the listed requirements for investigational new drug applications, and it complements ICH Q2, and it applies to method development and validation of in-house developed methods, as well as for verification of compendial methods and for transfer of analytical procedures. The guide states that you can use a different approach than the recommendations in the guidance, but then you should discuss the matter with appropriate FDA product quality review staff before you submit your application to the FDA, which is always a good thing to do. And it also demonstrates method equivalency, so this is also important. So now we go to the next slide, and this talks about the objective and the background of the guidance. The objective of the guide is to provide recommendations on how the industry can submit analytical procedures and methods validation data to support the documentation of the identity, the strength, the quality, purity, and potency of drug substances and drug products. The reason for method validation and related documentation are regulations like 21 CFR 314, 601 to 11, with statements such as each NDA and ANDA must include the analytical procedures which are necessary to ensure the identity, the strength, the quality, the purity, and the potency of the drug substance and also the drug product. So this is also very important. Okay, what else is important here? We go to the analytical methods development here. The chapter on analytical methods development is new compared with the existing guidance. There is a lot of emphasis on robustness testing. I mentioned it already earlier on. That should be evaluated during early stages of development because results will influence its ideal technique and parameters. This is in line with ICH-Q2, which also recommends conducting robustness testing during method development. And FDA recommends systematically adopting a systematic approach for robustness studies, for example, design of experiments, DOE, which also is something which we know from the QPD principles, and this is being done for critical method parameters. Criticality should be established with initial risk assessment, and operational limits should be evaluated with multivariate experiments. I will show you later on a little bit more into detail how to fit all this together. And the last bullet here is very important, and this created more work for the industry. Development data should be submitted within the method validation section if they support the validation of the method. So I think the FDA would like to see the development data related to method validation, and again, if they only if they support the validation of the method. I think which is basically nothing new because also method validation in the past or currently is requiring to be done. The experiments should be done under GMP conditions, and so it's basically the same like now. And even if the method validation, the method development has been done, method validation has been done during development, I think FDA still can ask for those documentation to be done under GMP conditions. So this is also very important. Now we talk about, so I think this is quite clear. Initial risk assessment is important, followed by multivariate design of experiments. Data should be submitted within the method validation section if they support the validation of the method, only if they support the validation of the method. Now here I have a couple of examples and the definition for robustness testing, which is very important. Robustness has been defined as a measure of the method.
Thank you so much for taking a few minutes to watch this clip from one of our online meetings. Please don't forget to subscribe to our channel, stay up to date and stay compliant. This video is brought to you by GRCTS. We are also known as GRC Training Solutions. And here's a quick information about us and what we do. We conduct many compliance and non-compliance training courses throughout the year, covering some of the most important areas within several industries. Our focus is to provide the best and the most up-to-date classes to our audience. All training modules are designed and facilitated by industry subject matter experts who possess years and years of experience and understand compliance very well. Our customers attend our online classes from all over the world. If you're not available to attend the live meeting classes, you can always opt in to get unlimited access to our on-demand or recorded classes. We also provide in-person and face-to-face workshops, on-site workshops where our experts will visit your company at your location and train you and your colleagues, LMS trainings, and finally corporate subscription plans, where you pay a little amount to enjoy unlimited access to our courses, we have monthly, quarterly, and annual subscription plans. Here's our contact information for you. If you're interested in learning in any of these topics, or if you know a colleague or a friend who might benefit from our classes, please contact us or visit our website for more information. It's www.grcts.com. You can always send us an email, support@grcts.com, or just give us a call, 248-233-2049.